Hello everyone, welcome back. Today we will be studying about nervous system. So basically in first part we will cover anatomy, physiology, and that is the basic things, and in later we will also cover medical, surgical, nursing. Okay, so let's start. Nervous system is one of the regulatory systems of body which controls all the activities of body quicker than the other control system in the body, like endocrine system. Okay, and it made up by a network of billions of neurons, right? Functional unit of nervous system. Then neuroglia or glia, the supporting structure of nervous tissues. It includes astrocytes, oligodendrocytes, and microglia into CNS, and Schwann cells and satellite cells into peripheral nervous system. Now first comes to astrocytes, largest and most numerous of the neuroglial cells. So it is the largest among all the neuroglial cells. It is the largest and most numerous number. It is present, okay, and it helps to make BBB. What is BBB? That is blood-brain barrier. Fine, it is clear. Now come to next, that is oligodendrocytes. It responsible for formation and maintenance of myelin sheath around the CNS axons. So first one was working as a BBB, blood-brain barrier. Now this one is making and maintaining myelin sheath around CNS axon. Next is microglia. Okay, what is microglia now? Cells function as macrophages which works for phagocytosis when they migrate to damaged CNS tissue. So basically when microglia is migrating to damaged CNS tissue, they are working as macrophages which help in phagocytosis. So hope up to this it is clear. Now comes to satellite cells. It enclosing the cell bodies of sensory neurons in spinal ganglia. Now Schwann cell, what is that? It forms myelin sheath on neurons of PNS. Fine. Oligodendrocytes helps in forming myelin sheath around CNS axon, and Schwann cells it helping to form myelin sheath on neurons of peripheral nervous system. Okay, so next, neuron or nerve cell. Neuron is the structural and functional unit of nervous system, and like muscle fibers, neurons also have property to produce action potential. We know, right, that in muscle they are working by producing action potential, and same things happen with neurons too. They also have property to produce action potential, and duration of action potential is about 1 millisecond. Can you imagine that 1 millisecond is how, like, fraction of second? So within that much time they are working. That's why it is called the nervous system is the fastest. Like they work quicker than any other system in our body, or impulses in response to stimuli. For example, structure of neuron. Like any other cells in the body, neurons also having almost all the organelles in the cytoplasm except two main difference. That is, what are they? That is presence of process and absence of centrosome. The neurons, nerve cell have three parts. Now let's see one by one. Nerve cell body, what is that? Cell body contains a nucleus which is surrounded by cytoplasm. Now cytoplasm consists of lysosome, mitochondria, nasal bodies and Golgi. So next point is nucleus does not contain a centrosome. So the nerve cell cannot multiply like other cell. It is clear? Now comes to nasal body. So let's mark it up with another color, that centrosome is absent in nucleus, right? That's why they cannot multiply. And nasal bodies, what is that? First time described by Franz Nagel, are spotted like tiger, also called tigroid substance. Nasal body synthesis protein because it contains ribosome. So here who is producing, synthesizing protein, that is nasal body, because it contains ribosome. Now nasal body present into nerve cell bodies and dendrites but not into axons. Axon में present नहीं रहता है, बस वो cell body and dendrite में present है. Now comes to dendrites. Dendrites are main receiving or input region of neuron. It transmits information towards the nerve cell body, and it is a branched short process of neurons. So basically what is dendrites? So let's see in this picture. All these are dendrites. Can you see? Yeah. Like a star, it is our cell body, and this is our nucleus. And see, this is nucleus, this is cell body. Okay. So basically this is the dendrite. Hope you understood. So what they are working? Their main work is to receive information, right, towards the cell body. So they are taking information from outside to inside. Okay, now comes to axon. It is a large process of nerve cell. Can you see? This is the axon. Basically, this long axon. See, long process of nerve cell. The length of the longest axon is about 1 meter. So it will be about 1 meter. Axon is output region which convey impulses from cell body to another neurons. Dendrites was working as the input, right? But it is working as output because they are conveying impulses from cell body to another neurons. The central core of axon, exoplasm covered by membrane, that is axolemma, and membrane covered by a thick tubular lipoprotein, that is called myelin sheath. So the central core of axon, exoplasm covered by a membrane, axolemma, and membrane covered by a thick tubular lipoprotein sheath, that is called myelin sheath. Myelin sheath is absent at regular interval, and this absent area is called node of Ranvier. See, where is node of Ranvier? This one. What is myelin sheath here? Can you see? This black one they have marked. This is our myelin sheath. And what is Schwann cell now? It is also present here. We will study in later page. Let's see. Myelin sheath is responsible for the white color of nerve fibers and for faster conduction of nerve impulses through the nerve fiber. And surrounding the myelin sheath, there is a thin membrane called neurilemma sheath or sheath of Schwann, made up of Schwann cells. I was telling you, this one is Schwann cell. And sheath of Schwann is necessary for the formation of myelin sheath into PNS. In peripheral nervous system, these Schwann cells are forming the myelin sheath. And myelinogenesis of PNS start at 4 months. If anyone is asking you, like when in PNS, peripheral nervous system, this myelinogenesis process is started, that will be, answer will be 4th month of intrauterine life and completed by 2nd year of birth, 2nd year after birth, same as well. So comes to neurilemma. Is absent in CNS, so regeneration of nerve does not occur in CNS. But sometimes neuroglia or glia, it constitutes half volume of the CNS, and cells are responsible for myelinogenesis in the CNS. As neurilemma is absent in CNS, that's why the regeneration process cannot be happen in CNS. But sometimes what they are saying, neuroglia or glia in half volume, it present in CNS, and those are the responsible for myelinogenesis in the CNS. Now comes to next point. Distal end of axon divided into many branches called axon terminals. This is the axon terminals. If you see the picture and study, then you will remember. See, this is the terminals. Okay, I'm using different color so that you can understand. Okay, this one, this, can you see? So this is basically axon terminal, synaptic bulbs. Okay. Now, special note they have underlined. See, the dendrites and axon together called process or nerve fibers. So what is nerve fiber? That is exoplasm plus axolemma. Okay. Now classification of neuron or nerve cell. First one is depending upon structure. So let's see, myelinated neuron. This neuron covered by myelin sheath, and non-myelinated neuron, this neuron does not have myelin sheath, mainly into fetal life. Next is depending upon the distribution. What are they? Somatic and visceral. Somatic means pertaining to the body wall neuron. It supplies the skeleton muscles of the body. And in visceral neuron means internal organ, right? It will be autonomic because it is not under our control. This is voluntary muscle. First one. So somatic is voluntary and visceral is non-voluntary. It's supplied to the internal organ of the body. Next point, that is depending upon the sources of origin. What are they? Cranial nerve and spinal nerve. These nerves arise from brain. Total number is 12. Cranial nerve, so total number of cranial nerve is 12. And total number of spinal nerve is, that is 31. They arise from the spinal cord. Then depending upon the function. See, motor neuron that carries impulses from the CNS to either muscle tissue to stimulate contraction or to glandular tissue to stimulate secretion. This will be your motor neuron. So what are they? Let's revise it again. Neuron that carries impulses from CNS to either muscle tissue to stimulate contraction or to the glandular tissue to stimulate secretion. Basic funda is that. Now, sensory neuron, what are they? See, neuron that conducts sensory impulses toward the brain and spinal cord. So this will be called sensory neuron. So let's start synapse now. The junction between two neurons called synapse. So one neuron consists of dendrites, axon and cell body. You have seen, right? So another junction, so between two neurons, the junction it is there. So that will be called synapse. The neurons sending the signals called presynaptic neuron, and the neurons receiving the message, it is called postsynaptic neuron. The plasma membranes of presynaptic neuron and postsynaptic neuron in chemical synapses are close, but do not the minute space between presynaptic neuron and postsynaptic neuron called synaptic cleft, that is 20 to 50 nanometer. There is a minute space between these two neurons and it is called synaptic cleft. What is that? That is 20 to 50 nanometer. Now comes to neurotransmitter. The chemical substance responsible for the transmission of impulses through the synapses called neurotransmitter. Neurotransmitter is synthesized by the presynaptic neurons and stored in vesicles. Then neurotransmitter passes through synaptic cleft from presynaptic neuron to postsynaptic neurons and attached to receptor. After attachment, it transmits the impulses from one neuron to another neuron. Did you understand? So let's revise it again. The chemical substance that is responsible for transmission of impulse to synapses, it is called neurotransmitter basically. So first is, it is synthesized by presynaptic. We were studying in the last topic, right? Presynaptic and postsynaptic neuron is present. So basically neurotransmitter is synthesizing by presynaptic neurons and stored in the vesicles. Now how it will work? See, neurotransmitter passes through synaptic cleft. What is synaptic cleft? That is the gap between presynaptic and postsynaptic. Okay. Then it will passing from the synaptic cleft from presynaptic neuron to postsynaptic neuron and attached to the receptor. After attachment, it transmits the impulses from the one neuron to another neuron. Hope it is clear now. Now comes to next point. See, types, that is exciting transmitters. These transmitters are responsible for the conduction of impulses from the presynaptic neuron to postsynaptic neuron. Examples are like acetylcholine, noradrenaline, glutamate and histamine. You have to remember that what all are the exciting neurotransmitters, that are acetylcholine, noradrenaline, glutamate and histamine. Revised. Then next is inhibiting transmitter. So this transmitter, what they are doing, they are responsible for the inhibition of conduction of impulses from presynaptic neurons to the postsynaptic neuron. So you have to again remember the example: dopamine, gamma aminobutyric acid, that is GABA, and serotonin, that is 5-hydroxytryptamine. Okay. So basically first one is conducting the impulse, right? And next one is inhibiting the conduction. Hope it is clear. Now division of central nervous system. Primarily the nervous system is divided into two divisions, that is CNS, central nervous system, and PNS, that is peripheral nervous system. In next session we will be continuing central nervous system. Hope you have understood up to these points. So revise it once again and come back to next session. Thank you.